Hello, my name is Ellen Winesapple, and welcome to EpiCypher's CUTANA cut-and-run protocol video. This video will be broken up into 4 sections. First, I will give a brief overview of how cut-and-run works and review important considerations for experimental design and sample preparation. Then I will give a walkthrough of the protocol. Next, I will go over protocol variations and wrap up with a few frequently asked questions. Cut-and-run stands for cleavage under targets and release using nuclease. Concanavalin A or Con-A beads coated with lectin proteins are used to bind the glycoconjugates found on the membranes of cells or nuclei. The membrane is permeabilized and chromatin is flagged with antibodies for the target of interest, in this case to a histone PTM. Next, protein AG micrococcal nuclease, also known as PAG eminase, is added, which binds to antibody-bound chromatin. The addition of calcium chloride activates the enzyme's cleavage of the target complex, which is then able to diffuse out of the cell. The DNA is collected from the supernatant and purified before library preparation. Experimental success is tested with a Bioanalyzer trace before sequencing with either a sequencing core or a benchtop sequencer. When the results are in, you can analyze and view your data with integrated genome browsers or other appropriate visualization tools. Our cut-and-run protocol provides a validated platform for genomic profiling of histone PTMs, transcription factors, chromatin remodelers, writers, and readers. If you are new to cut-and-run, going after new targets, or testing new antibodies, it's important to carefully consider the cell types, experimental conditions, controls, and antibodies that will best address your biological question of interest. Our protocol is compatible with multiple cell and sample types. Native or unfixed cells are ideal if you are profiling histone PTMs and transcription factors. These cells can come from various sources, including adherent and suspension cell lines, as well as tissue samples. The CUTANA cut-and-run protocol also works with lightly cross-linked cells. Sample fixation is recommended for profiling transient chromatin interactions and certain subsets of histone PTMs. However, you can also use nuclei, which is useful when mapping rare PTMs or proteins. We also recommend using nuclei when profiling immune cell populations, since the Con-A beads can activate some immune cell types. Lightly cross-linked nuclei can also be used, and as a final note, all sample inputs can undergo cryopreservation. Adapted workflows for the various sample inputs can be found in the Protocol Variations section of this video. For those new to cut-and-run, we recommend starting with at least 500,000 cells per sample, and then titrating cell inputs to determine the requirement for your specific target and cell type. EpiCypher's cut-and-run protocol, without any adaptations, is optimized from 500,000 to as few as 5,000 cells without compromising data quality. Controls are an essential part of experimental design. Appropriate controls are key to a successful cut-and-run experiment and should always be run in parallel with your experimental samples. For a positive control, we recommend treating samples with a validated antibody, such as our SNAP-ChIP certified H3K4ME3 or H3K27ME3 antibodies, which generate consistent peaks in cut-and-run. Rabbit IgG antibody can be used as a negative control, which will reveal the level of background signal. We also suggest using spike-in controls, such as E. coli DNA for normalizing sequencing data. These careful considerations in experimental design will allow you to easily evaluate the success of your cut-and-run experiment and perform downstream data analysis. Before getting started, make sure you have access to all the required reagents and equipment. PAG eminase, Con-A beads, and antibodies need to be carefully handled. Our protocol has been adapted for high-throughput sample preparation and processing in an 8-strip PCR tube format, offering consistent sample handling and reproducibility in cut-and-run experiments. It is important to use the molecular-grade buffer ingredients listed here. For a complete list of all materials and our recommended vendors, click Show More in the video description below. Cut-and-run has 6 main working buffers, many of which are used to make the next buffer on the list. I will go over them fairly quickly, so feel free to pause the video to take a closer look. If working with nuclei or cross-linked materials, alternative buffers and preparations are required, which will be addressed in the Protocol Variations section of this video. Since the cut-and-run protocol is in an 8-strip tube format, we have provided you with the 8x buffer calculations and a scaling table at the end of this section, all with extra volume calculated in for pipetting error. However, we always recommend validating the volumes with final concentrations. The pre-wash buffer is not used in the protocol, but is a stock you pre-make and use to prepare your wash buffer. Filter sterilized buffers using a 0.2 micron filter. Pre-wash buffer can be stored up to 6 months at 4 degrees Celsius. We prepare a 10x stock from a mini protease tablet just prior to making the wash buffer. A large portion of the wash buffer will be carried over to make the digitonin buffer, and the rest will be used in the protocol. Before getting started with the digitonin buffer, if you are using cells other than K562, MCF7, or A549, you should empirically determine the optimal digitonin concentration for your specific cell type. The digitonin buffer will need to be made fresh for each experiment. Once combined, some will be carried over for the antibody buffer, while the rest can be stored overnight for the day 2 portion of the protocol. Use the carried over digitonin buffer and EDTA to make the antibody buffer. We recommend using this buffer to dilute your antibody stock. Diluted antibodies can be stored at 4 degrees Celsius for 1 month, but the antibody buffer will need to be made fresh for each experiment. Bead activation buffer can be pre-made, filter sterilized, and stored for up to 6 months at 4 degrees Celsius. Place aliquots on ice when it's time to activate the Con-A beads. The stop buffer can also be pre-made in larger batches, filter sterilized, and stored at 4 degrees Celsius. You will need 33 microliters per sample to stop the PAG emanase digestion. We have also provided the scaled-up volumes for these buffers. We always recommend using final concentrations to validate your volumes. In addition, please keep in mind that these volumes are adjusted to the nearest decimal and are pre-calculated to include dead volume. We are now ready to begin the experiment. In the first section of the protocol, we strongly recommend performing batch bead activation, which will be shown in this walkthrough. Batch processing is performed in a 1.5 ml tube and thus requires an accommodating magnetic rack. If you only have an 8-strip tube magnetic rack, bead activation can be performed in 8-strip tubes, but you may experience more bead loss, clumping, or drying. Don't forget to keep your bead activation buffer on ice. Resuspend the Con-A bead stock by gently vortexing on a 6 or 7 setting, or gently inverting the tube a few times. Give the tube a quick spin to remove any material that might be in the cap. Transfer the appropriate amount of Con-A beads for your number of samples to a 1.5 ml tube. Place the tube on a magnetic rack until the slurry completely clears. Take care not to touch or disturb the beads as you remove and discard the supernatant. Resuspend for 30 seconds with 100 microliters per sample of cold bead activation buffer. Place the tube on the magnet until the slurry clears and carefully remove the supernatant. Repeat this process for a second wash. After removing the supernatant from the final wash step, add 11 microliters per sample of cold bead activation buffer and resuspend with a 200 microliter pipette or by gently vortexing. If you notice any residual liquid on the walls of the tube, perform a quick spin to minimize bead loss. You can either proceed to the next section in the 1.5 ml batch processing format or the 8-strip tube format. If you are proceeding in an 8-strip tube format, aliquot 10 microliters of activated Con-A bead slurry to the bottom of each tube. Keep the beads on ice as we proceed to section 2 of the protocol. Here is an overview of section 1. If working with inputs other than native cells, please see the protocol variations section of this video before proceeding with section 2. Here are the required materials for this section. Please make sure your wash buffer is at room temperature to avoid shocking your cells. We are shooting for around 500,000 cells per sample, so you will need 4.4 million total cells per 8-strip tube to account for potential bead loss at wash steps. You will also want to determine the percentage of viable cells, which should be greater than 95%. Transfer cultured cells to a collection tube and centrifuge for 3 minutes at 600 G at room temperature. Then decant or pipette off the culture media. Add 100 microliter per sample of room temperature wash buffer and resuspend the pelleted cells for 30 seconds gently yet thoroughly with your pipette to avoid cell lysis. Centrifuge for 3 minutes at 600 G at room temperature to repellet your cells. Then remove the supernatant. Again, resuspend with wash buffer, spin, and remove the supernatant to complete your second wash step. Your final resuspension is with 100 microliters per sample of wash buffer, taking care to avoid cell lysis. It's now time to combine your cells with the Con-A beads. If you continue batch processing at this step, then add 10 microliters of beads per sample to the 1.5 ml tube. If your beads are already in 8-strip tubes, transfer 100 microliters of resuspended cells to each tube. Gently vortex or pipette to mix. If the consistency looks good, incubate the cell bleed slurry at room temperature for 10 minutes, during which time you can prepare for Section 3. Here's an overview of Section 2. For Section 3, you will need these materials. Make sure you include samples treated with a positive and negative control antibody, in addition to experimental samples labeled with the antibody for the target of interest. We will walk through batch processing and then show the same workflow summary for the 8-strip tube format. At the end of the 10-minute incubation, it is normal to see settling in the tube. Place your 1.5 ml tube on the magnetic rack and remove the supernatant. Add 55 microliters of cold antibody buffer per sample if batch processing and resuspend quickly to prevent bead drying, but gently to prevent cell lysis. At this stage, if you had been batch processing, it's now time to aliquot 50 microliters of your slurry into 8-strip tubes. We are now on the 8-strip tube format. You will need to include positive and negative controls in every experiment. Add 0.5 microliter of antibody at a 1 to 10 dilution for the negative control, and a 1 to 100 dilution for the positive control and target of interest antibodies. Make sure to check your pipette tips to ensure the small volume was transferred. Gently vortex your strip tubes, and then place them propped up on a nutator as shown here to keep the beads in solution and away from the cap. Incubate your samples overnight at 4 degrees Celsius for a total of 16 hours. This is an overview of the Day 1 portion of Section 3 in batch processing. And here's the workflow in the 8-strip format. Remember to store the digitonin buffer at 4 degrees Celsius overnight to use on Day 2. In the morning, continue with Section 3. Get the digitonin buffer and pour it into a buffer reservoir on ice. You should also prepare for the next section by retrieving the tube of PAG-eminase and placing it on ice. Obtain your samples from the nutator and give them a quick spin with the caps facing in. Place the 8-strip tubes on a magnetic rack and wait for the slurry to clear. Remove your supernatant and without removing the tubes from the magnet, add 250 microliters of cold digitonin buffer. Repeat this process for a second wash, again without removing the tubes from the magnet. Remove the supernatant from the final wash, taking the tubes off the magnet rack, and resuspend in 50 microliters of digitonin buffer by gently pipetting or vortexing. It's common for beads to be clumpy at this step, so be sure to thoroughly disperse. Here's a summary of the Day 2 portion of Section 3. You will want to move quickly to Section 4. Make sure you have all of your materials for this section. PAG-eminase and digitonin buffers should be kept on ice. Add 2.5 microliters of PAG-eminase to each sample and start a 10-minute incubation at room temperature. When the incubation is over, give your samples a quick spin, again with the caps facing in, before placing them on the magnetic rack. Once the slurry clears, remove the supernatant. While keeping the tubes on the magnet, add 250 microliters of cold digitonin buffer to wash the bead pellet. Remove the supernatant and repeat for a second time. When the supernatant from the final wash is removed, you may remove tubes from the rack and resuspend with 50 microliters of digitonin buffer. Again thoroughly, resuspending any clumps. Set your tubes on ice. Here's an overview of Section 4 of the protocol. For Section 5 of the protocol, you will need these materials. Leave the tubes on ice as you add 1 microliter of calcium chloride per sample. This is a critical step, as the calcium chloride activates the cleavage activity of the PAG-eminase enzyme. Resuspend by vortexing and place the tubes on a nutator for 2 hours at 4 degrees Celsius, with caps slightly elevated. Before the incubation ends, prepare the thermocycler settings and pour the stop buffer into a buffer reservoir. When the 2-hour incubation is over, retrieve your tubes, giving them a quick spin with the caps facing in. Add 33 microliters per sample of stop buffer. If you are using spike-in DNA for normalizing sequencing data, aim for 0.5 to 5%, ideally close to 1%, of the total read counts of the sequencing data. You can either pre-mix the spike-in DNA with the stop buffer or add the appropriate optimized amount directly to the tubes. Place the tubes in the thermocycler for 10 minutes at 37 degrees Celsius. During the incubation, you should prepare your columns and collection tubes for DNA purification. When the incubation is complete, place your tubes on the magnetic rack and transfer each of your supernatants into a new tube. At this point, you should follow the instructions for your DNA purification kit. Here's an overview of Section 5. In this section, we will summarize the steps for eluting DNA, for library preparation, and quality control steps prior to sequencing. We recommend eluting DNA in 10 microliters of elution buffer. Carefully quantify 1 microliter of eluted DNA using the Qubit fluorometer. Here are some example yields for common targets from 500,000 native K562 cells. We expect about 200 nanograms for our H3K27ME3 positive control antibody, while transient factors like SMARCA4-BRG1 yield about 15 nanograms. Once your samples are quantified, you can proceed to library preparation. Do not assess fragment size distribution prior to library prep, as the yield may be below the limit of detection for this analysis and is not indicative of experimental success. Described here is our standard PCR conditions for cut-and-run library preparation. Please refer to our online protocol for modifications in library preparation when working with transcription factors, including those with expected fragment sizes of less than 120 base pairs. Examine your experimental success using a bioanalyzer or equivalent approach before proceeding to sequencing. Depicted here are bioanalyzer traces for negative IgG and 2 of our positive control histone PTM antibodies. Successful experiments show that specific fragment size distribution around 300 base pairs, which correlates to a mononucleosome plus a 125 base pair adapter. Fragment size distribution as in the IgG negative control indicates nonspecific cleavage. Once you have obtained the specific cutting pattern for your target factor, proceed to sequencing. You can multiplex several samples in one lane, as you will only need between 3 to 5 million reads per sample. See EpiCypher.com/protocols for more information. In the next section of this video, we will go over experimental variations of the cut-and-run protocol, including for nuclei extraction, cross-linking cells, cryopreservation, and adherent cell handling. Cut-and-run is compatible with nuclei, which is especially valuable when working with immune cells. We recommend using nuclei with immune cells since the lectins on CAN beads may inadvertently stimulate them during binding. Our PAG eminase is able to diffuse into the nuclear pores, alleviating the need for membrane permeabilization. But keeping 0.01% digitonin in the workflow will prevent CAN beads from sticking to the tubes. As shown here, you will need to make a pre-nuclear extraction buffer that should be filter sterilized and stored at room temperature for 6 months. This buffer is not used in the protocol itself, but instead is used when creating the nuclear extraction buffer. Make the nuclear extraction buffer as described here. Filter sterilize and store at 4 degrees Celsius for 1 day. This buffer will be used for nuclei extraction and resuspension in the beginning of Section 2 of the protocol. We have provided the scaling chart for your convenience, and volumes are pre-calculated to the nearest decimal and are adjusted to include dead volume. The beginning of Section 2 is the only necessary adaptation when extracting nuclei. Harvest 0.5 million cells per sample by spinning for 3 minutes at 600 G at room temp. Wash cells in 100 microliters per sample, room temperature 1x PBS, and spin for 3 minutes at 600 G at room temp. Resuspend cells in 100 microliters per sample of cold nuclear extraction buffer. After resuspension, incubate samples on ice for 10 minutes, then spin for 3 minutes at 600 G at 4 degrees Celsius, and decant or pipette to remove the supernatant. Finally, resuspend nuclei in 100 microliters per sample of cold nuclear extraction buffer, and proceed as normal at Step 7 of Section 2 to bind nuclei to the CAN beads. Cut-and-run is compatible with cross-linked cells or nuclei, But it is important to remember that typical chip cross-linking is not compatible with cut-and-run. We recommend a light cross-linking using 0.1% formaldehyde for 1 minute to stabilize label targets such as acetylhistone marks. Alternatively, medium cross-linking at 1% formaldehyde for 1 minute may also be used. It's important to determine the biological impact of cross-linking by titrating the amount and duration of cross-linking in your experiments. We have found that heavy cross-linking, as used in ChIP, which uses 1% formaldehyde for 10 minutes, significantly reduced DNA fragment recovery in cut-and-run and is not recommended. You will need to make a cross-linking specific pre-wash buffer that will make the basis for subsequent cross-linking buffers. To make the cross-linking pre-wash buffer, use the earlier described normal pre-wash buffer as the base, with a final concentration of 1% Triton and 0.05% SDS. Filter, sterilize, and store this buffer at 4 degrees Celsius for up to 6 months. Here's the scaling chart of the normal pre-wash buffer and the cross-linking pre-wash buffer. The cross-linking pre-wash buffer is used in place of the normal pre-wash buffer to make a cross-linking specific wash buffer. The subsequent digitonin and antibody buffers will also be modified for cross-linking. Do not use the modified buffer preparations to dilute your antibodies. Instead, you will need a normal antibody buffer stock for diluting your antibodies. For cross-linking, there are 2 main changes, one at section 2 and the other at section 5. Note that there is an extra overnight incubation to reverse the cross-links. At the beginning of section 2, add formaldehyde directly to the cell culture for a final concentration of 0.1%. Gently vortex cells and incubate for 1 minute. Quench by adding glycine to a final concentration of 125 millimolar and then gently vortex to mix. Spin cells for 3 minutes at 600 G and resume as normal at step 7 of section 2 to bind cells to activated Can beads. After completing step 28 of section 5, you will need to reverse crosslink by adding SDS to a final concentration of 0.09% and adding proteinase K at 20 micrograms per microliter. Incubate overnight at 55 degrees Celsius in a thermocycler and resume as normal at step 29 of section 5. Cryopreservation and adherent cell preparation are 2 additional variations that may need careful handling. Cryopreserve cells in 100 microliter aliquots in respective growth media supplemented with 10% DMSO. Slowly freeze samples in a -80 degrees freezer. Avoid snap freezing in liquid nitrogen, as this will result in cell lysis and chromatin leakage, which contributes to higher backgrounds. When ready to use samples, quickly thaw cells at 37 degrees Celsius in a thermomixer before proceeding with section 2 of the cut-and-run protocol. When working with adherent cells, avoid using trypsin, as this will degrade glycoproteins needed to bind the Can beads. Instead, we recommend gently scraping adherent cells while in culture media and monodispersing them gently using a pipette before proceeding directly to cut-and-run wash steps. The final section of this video will cover answers to some frequently asked cut-and-run questions. Cell permeabilization is critical for PAG-MNase to diffuse into the cell. To test that permeabilization was successful, you can use trypan blue staining. Another issue that researchers sometimes experience is bead clumping. First, avoid freezing beads in the -20 degrees Celsius freezer and only store them at 4 degrees Celsius. Clumping is normal and can be dispersed by pipetting or gently vortexing. If you are unsure, you can check binding efficiency by staining. Choosing your antibody is one of the most important decisions in a cut-and-run experiment. Our team has found through repeated testing that many ChIP antibodies, including heavily cited ones, are non-specific and have low efficiency pull-down. We recommend using EpiCypher's SNAP ChIP certified and cut-and-run compatible antibodies. Check the link below for more information. Though our PAG-MNase is expressed in E. coli, there are only trace amounts of residual E. coli DNA fragments present in the PAG-MNase prep. We suggest spiking in E. coli DNA to provide sufficient read depth for sequencing read normalization. We have tested several DNA isolation techniques and found that kits provide the best and most consistent results. However, phenylchloroform and ethanol precipitation approaches will work too. In ChIP protocols, it is common practice to analyze isolated ChIP DNA using agarose gel or capillary electrophoresis to assess fragment size distribution prior to library preparation. However, this is not recommended for cut-and-run, since the amount of DNA is likely to be below the sensitivity of detection for these approaches. Instead, we recommend checking your DNA on a Bioanalyzer or TapeStation after library preparation. You should expect to see approximately 150 to 300 base pair nucleosomal peaks for histone PTMs. For transcription factors, fragments less than 120 base pairs are generated. You also need to keep in mind the adapter length of about 125 base pairs. The amount of DNA generated can be variable depending on antibody specificity, efficiency, and target abundance. In general, you should expect 10 nanograms or more recovered DNA. To enrich for fragments less than 120 base pairs, reduce the inactivation temperature after end repair from 65 degrees Celsius for 30 minutes to 50 degrees Celsius for 1 hour. In addition, after the ligation reaction, the ampere bead size selection ratio should be increased from 1.1x volume to 1.75x volume. Otherwise, follow our standard protocol and use 5 to 10 nanograms per reaction. Lower amounts may lead to increased adapter dimer. Results from challenging cell inputs or targets may be ambiguous, so EpiCypher recommends including positive and negative controls into every experiment. If the QC checks for the positive control perform as expected, then you can proceed to sequencing. If sequencing results for challenging cell inputs and targets are not satisfactory, further optimization may be necessary. The low sequencing depth requirements of CUT&RUN makes it possible to run multiple samples on benchtop sequencers. Typically, you will only need between 3 and 5 million reads per sample. The bioinformatic tools needed for analysis include quality control, mapping, and visualization software. To interpret the final sequencing data for experimental success, compare the sample tracks with the positive and negative control tracks. Your samples should give a distinct peak that passes a defined threshold limit and is translated to be biologically meaningful or validated. We have now reached the end of the CUTANA CUT&RUN protocol video. We have compiled additional resources in the video description below for those who would like to learn more about CUT&RUN. This video was made possible through efforts of our science, sales and technical support, and marketing teams, as well as significant contribution from Solomon Hailu, who also animated and edited the video. If you have any questions about your CUT&RUN assay or would like to purchase supplies for your CUT&RUN experiment, please reach out to us on social media, through our website, or email us at info@epicypher.com.